Hello, everybody, and welcome to week 26 of Human Circulation and Gas Exchange. Today, we are going to be looking at angiology, and specifically within the study of blood vessels, we're going to be looking at the structure and function of them. In terms of this week's learning outcomes, we are going to be looking at the following. So, by the end of today's virtual lesson, we'll all be able to describe the structure and function of blood vessels via exploring the anatomy and physiology. And for learning outcome 2, apply what you have learned via relating the structure of blood vessels to the function. Essentially speaking, completing pages 7 and 8 of your e-workbooks. So this is an overview of our weekly schedule. We are currently on week 26. We have got one more week before April vacation, so next week we'll be looking at the components of blood and the structure and function of a red blood cell, and then that ultimately would comprise of 5 more weeks before ultimately the final teaching. So in terms of a recap of last week's topic, which explored circulatory systems, here is an overview of the pulmonary and systemic circuits. So in green, you can identify the systemic circulatory system, and then within the tan colour is the pulmonary circuit. As you can see, the pulmonary circuit is transporting blood from the right ventricle to the lungs for ultimately carbon dioxide to be removed and oxygen to be absorbed, and is then transported to the left atrium. The systemic circuit then ultimately comprises of the left ventricle, and ultimately that then pumps blood to the rest of the body to provide all its bodily tissues with oxygen, nutrients, whatever it might be. And then once carbon dioxide is produced, your veins ultimately transport it to the right atrium, where the process and the cycle repeats. So this is an overview of systemic circulation. So as we've mentioned, the systemic circulatory system carries oxygen and nutrient-rich blood to bodily tissues and removes carbon dioxide and other waste from the tissues. All systemic blood vessels branch from the aorta. Deoxygenated blood returns to the heart through the systemic veins, which in turn drains blood into the right atrium of the vena cava. The pulmonary circuit is ultimately carrying deoxygenated blood from the right ventricle to the lungs to allow gas exchange within the alveoli. That's something that we'll look within a couple of weeks' time. This is then exchanged to provide oxygen-rich blood, which is then transported to the left atrium via the pulmonary vein. There are other circulatory systems within our body. We've got what is called the hepatic portal circulation, which carries nutrient-rich deoxygenated blood from the gastrointestinal organs and spleen to the liver and mixes with oxygenated blood received from the hepatic artery. This blood then leaves the liver through the hepatic veins, which is then drained into the inferior vena cava. We have then got what's called fetal circulation. Now this will only apply to those that can ultimately give birth, so generally speaking, women. The circulatory system of a fetus, otherwise known as fetal circulation, is the exchange of materials between the fetus and the mother itself through the umbilical cord and placenta. So today's topic, essentially speaking, is exploring the structure and function of blood vessels. So ultimately, what are the purpose of blood vessels? The main purpose of blood vessels within the body, contributing what is known as homeostasis, provides the structures for the flow of blood to and from the heart. They transport oxygen and nutrients to bodily tissues, as well as remove carbon dioxide and waste from them. And to kind of put this into perspective, the heart could be compared to a major train station or train stations, and the connecting railway lines are the blood vessels. So what are the different types of blood vessels? Well, there is ultimately 5 main types of blood vessels. We've got arteries, arterioles, which literally means small arteries, capillaries, venules, small veins, and veins. Each of these types of blood vessel will have a different structure and function, and ultimately this is what we need to be exploring. Another question for you, and the question is, if blood is red, why are veins blue? Blood is always red, just different shades. Oxygen-rich blood is bright red, so that is number 1 on the image to the right of the screen. Oxygen-deprived blood is dark red, that's comprised by picture identified as number 2. Veins look blue because light has to penetrate the skin to illuminate them. Blue and red light, being of different wavelengths, penetrate with different degrees of success, and only blue light makes it back to our eyes. So that ultimately is why the veins look blue. Your lips almost may turn blue or pale if you're deprived of oxygen, what is known as cyanosis, because of lack of oxygen-rich blood running through the arteries and capillaries. In terms of the similarities and differences of blood vessels, what I want you to look at is briefly pause this screen and try and explore what is different between these types of blood vessels. So we've got an artery labelled A, vein labelled B, and capillary labelled C. What is different between all of those in terms of the structure? That is now what we're going to explore. This identifies what's called a pressure gradient of blood vessels. So this ultimately shows how arteries, which are in the red, ultimately is at higher pressure. And as the blood flows further away from the heart, ultimately pressure decreases. Arteries generally transport oxygen almost entirely, apart from the pulmonary arteries and pulmonary veins, which do the opposite. The capillaries are the site of exchange, and then we've got the veins. So as you can see, the pressure is highest within arteries and lowest within veins. So this will link ultimately to the structure and function. Now the basic structure of a blood vessel, it consists, generally speaking, of 3 layers, but it might not have 3 layers. Specifically, we'll look at a capillary that only has ultimately 1. But each of arteries and veins, generally speaking, have 3 layers, what's called the tunica interna, tunica media, and tunica externa. Very posh terms, they basically just mean an inner tunic, a middle tunic, and a outer tunic. And generally speaking, these layers are composed of different tissues. So within the tunica externa, generally speaking, it'll have an inner lining. The tunica media will have a muscular and or elastic middle lining. That might be more or less depending on the blood vessel. And a connective tissue outer covering for arteries and veins. A capillary doesn't have that, and that's linking specifically to its function, but we'll explore that shortly. So when we're looking at these different tunics, the internal, external and middle tunics, can you now identify the differences between the structures of all 3? And that ultimately will help you look at the similarities and differences of the structure. So, arteries. Because arteries were found empty at death, post-mortem in ancient times, they were thought to contain only air. However, we now know that arteries carry and transport blood, a liquid substance. The wall of an artery has 3 layers. It has a thick muscular and elastic middle layer which allows it to stretch easily or expand without tearing due to blood being at its highest pressure. So when we looked at the pressure gradient earlier, the arterial pressure was much higher than the pressure within a vein. Ultimately, you don't want an artery to burst, that would be catastrophic. Therefore, ultimately, that's linking to structure and its function. Arterioles are literally small arteries, and they are microscopic that regulate blood flow into the capillaries, ranging from 15 to 300 micrometers in size. The micrometer is 1/1000th of a millimeter in size, so these are very small. Veins. The structural changes of veins are distinct as they are in arteries, so ultimately they look slightly different, linking to the function. They are composed of essentially the same three layers, similar arteries, but the relative thickness of these will be different. They're under less pressure. Many have valves to prevent the backflow of blood. The pressure is low, so it needs help ultimately to help the blood shunt back to the heart, and it can do that ultimately by valves and also the contraction of skeletal muscles to help shunt the blood further up to the body to try and beat gravity. And similar to arterioles, venules are microscopic veins that receive blood from the capillaries and begin the return flow of blood back towards the heart. They are also microscopic. Then we have capillaries, and these are the smallest type of blood vessel in our body, and they have diameters of 5 to 10 micrometers. Red blood cells have a diameter of 8 micrometer, so 1/1000th of a meter, and have to be flexible in order to pass through the capillaries in a single file. So ultimately, when we're looking at the structure and function of a red blood cell, ultimately the structure of a red blood cell, it has to be flexible, otherwise it would burst to get through a capillary that is only 5 to 10 micrometers in size. They don't have an outer or middle membrane, otherwise substances will not be able to transport through blood vessels effectively. However, it does have an inner layer composed of epithelial tissue and a basement membrane which basically connects the capillary to the tissue site. So, in terms of today's task, you ultimately need to describe the structure and function of blood vessels and also apply what you've learned via creating a response ultimately in your e-workbooks. So learning outcome 1, to set the scene behind the task, is using literature on Google Classroom under week 26 titled Literature, the Structure and Function of Blood Vessels, as well as conducting your own research from the own sources that you've already got. Complete the following. So under week 26, open the document that is titled Formative Assessment, the Structure and Function of Blood Vessels. Using the Google Doc worksheet that is assigned to you, describe the structure and function of arteries, veins and capillaries, and also ensure that you are citing and referencing your sources. Once you have done that, I want you to submit and hand that in for feedback. And if you don't know how to access this document, there's some guidance on the following slide. So in order to access this Google Doc, you need to locate it. So once you've clicked on Formative Assessment, the Structure and Function of Blood Vessels, you'll see a box titled Your Work that is identified in the green square, and that is assigned only to you. So each and every one of your peers will have their own. You cannot see theirs, they cannot see yours. Once you have completed this document, click Turn In, which is identified by the blue box. That'll send it in, very similar to how you'd send an assignment within your e-portfolios. This is formative, so ultimately this is for me to check your progress in class. This isn't going to go towards your assignment as such. And from there I'll be able to provide you with feedback that will then inform you providing a response for your summative assessment. And then learning outcome 2 is what is ultimately going to inform your summative assessment. And ultimately you need to complete pages 7 and 8 of your e-workbook, which is relate the structure and function of blood vessels to the function. These are arteries, veins and capillaries that you need to do. And then ultimately utilise the worksheet that you've completed during learning outcome 1 to help you complete this task. Next week we are going to look at haematology. We are going to look at the components of blood and the structure and function of a red blood cell. And these are the references ultimately that we have used within today's session. As this is going online for copyright, I've just made sure that ultimately I've sourced and identified what sources I've used and where they are from. That is everything for today's session. Now it is a case of applying learning outcome 1 and learning outcome 2, and I will be on Google Hangouts and also email if you've got any questions.